Good evening, everyone, and thank you, Kathrin, for the warm welcome. As Kathrin said, my name is Justine Watkins. I'm a PhD candidate from Utrecht University, and today I'll be talking to you about the botulinum neurotoxin testing, specifically the transition that is ongoing. So Kathrin already gave an introduction to me, so I won't go over this slide, but I will say that I'm originally from New York, so that's why I have the North American map here. And I have a biological background, so that's definitely great to know. So my perspectives are coming from the background of a biologist, but now my PhD is definitely interdisciplinary, dealing with also the social sciences. So to give a little outline of what today's webinar will be about, I first would like to set the scene and then give a brief history of botulinum neurotoxin, and then go a little bit into the science, so looking at the mechanisms of toxicity, the different kinds of tests that are used. Then I will talk about an analysis of the transition, and then give some insights and highlights from this analysis. And then finally, I'll end with some takeaways and possible solutions. So botulinum neurotoxin, or BoNT, is a protein produced by Clostridium botulinum bacteria, and it causes muscle paralysis by blocking nerve signals at the neuromuscular junction. While BoNT is used medically to treat conditions like muscle spasms, it has also become widely known for its aesthetic applications, most famously under the brand name Botox, which reduces facial lines and wrinkles. To avoid confusion, I'll use the term BoNT products to refer to all formulations, not just the specific brand, which is known as Botox. What is less known, especially by the public, is that unless a manufacturer has implemented an approved alternative method, each batch of BoNT is still tested in mice, using the Mouse Lethality Bioassay to verify potency. This traditional assay can involve hundreds to thousands of mice each year worldwide to meet regulatory safety requirements in Europe, the UK, and beyond. I'll discuss this testing process and efforts to replace it with alternative methods during this webinar. Public concern over hidden animal use in BoNT testing led the ECEAE to launch the Stop Botox Animal Testing campaign in 2009. So you can see that here in this picture. They basically urged industry and regulators to adopt non-animal methods. After an international meeting hosted in the United States, in 2012, Allergen, which is one of the companies, became the first company to have a cell-based potency assay approved by both but in EU and in the USA for BoNT testing. However, the use of BoNT potency testing continued across Europe and the UK, prompting a new ECEAE petition, which is over here. So the European Coalition to End Animal Experiments was still fighting for the ending of animal testing. And this picture was taken in 2023, which basically shows that it's still ongoing, even 10 years after Allergen has actually phased in their own cell-based assay. This case captures the intersection of science, regulation, ethics, and public pressure. Issues we'll unpack as we continue to go through this topic. But to take us forward, I would first like to go back in time. Between 1817 and 1822, Justice Kerner, who's shown on the left, was the first to describe the toxin. He noted it as a foodborne paralysis originating from a fat poison found in spoiled sausages. This phenomenon was later known as botulism. In 1895, Emil van Ermingem isolated the responsible bacterium, Clostridium botulinum, from contaminated ham, confirming that the disease was caused by an anaerobic microorganism. By the early 1900s, scientists discovered that botulism could spread through more than just food. So at this point, we are assuming that botulism is only a foodborne illness, but as science progresses, scientists are realizing that botulism happens not just from food contamination. And today, we know there are 4 groups of C. botulinum. So if you look here, this is what C. botulinum looks like under the microscope. There are 7 known serotypes of C. botulinum, each affecting different species differently. For example, serotypes A, B, E, and F cause botulism in humans, while type D mainly affects mice. So this is just highlighting that different species react to the different serotypes differently. In 1946, researchers purified BoNT for the first time, opening the door to medical applications. It is important to note that Kerner, who is the discoverer of this toxin, had predicted its therapeutic uses before, but only a century later did this actually come to fruition. In 1960, Alan Scott, a Californian ophthalmologist who's pictured here, used BoNT injections to successfully treat strabismus, which is basically the misalignment of the eyes. This led to the development of the first BoNT product, Dysport, which was approved in 1990 in Europe to treat dystonias, which are hyperkinetic movements or involuntary muscle contractions. The product was proven to be effective for a number of muscle conditions and now has market authorizations in over 75 countries. In 1988 came the first documented use of BoNT for aesthetic treatments. In 2002, the FDA approved the use of BoNT for gallbladder lines, which are basically the frown lines here in our foreheads. In 2006, Germany was the first European country to authorize Azalor, which is it's a product name, for aesthetic use. And in 2009, it became authorized across Europe. So basically what I want you to take away from this little arrow, this little timeline, is that you see the rise in the aesthetic use of BoNT products from the 1990s up into the early 2000s. So really you're seeing the aesthetic use rising as we're learning that it can be used to treat wrinkles in the first place. Interestingly, independent of the way the product is used, BoNT is classified as a pharmaceutical and is thus only meant to be prescribed when the severity of these lines has an important psychological impact on the patient, which is highlighted in the quote seen here. So here it says, like I said already, that when the severity of these lines has an important psychological impact on the patient, only then should Azalor be prescribed. According to a survey by the International Society of Aesthetic Plastic Surgery, botulinum toxin remains the most common non-surgical procedure for both men and women and across all age groups, with more than 9 million procedures performed by plastic surgeons worldwide and a 26.1% increase, making it a topic of big proportions. As aesthetic consciousness in adults is continuing to grow, so is the use of aesthetic BoNT. So now that we kind of have an understanding of the societal relevance of BoNT, I would like us to get a little bit deeper into the science. So naturally, BoNT is a bacteria that is produced, naturally BoNT is a protein complex that is produced by the C. botulinum bacteria. So you can see this protein complex here. So this is what the protein complex looks like, and this is what the toxin is. And what you see on the screen is the BoNT-A complex, and it is what's most often used in pharmaceutical formulations. But it is important also to note that BoNT-B is also used, but I'll mostly be talking about BoNT-A as that's the most common. The way this protein causes paralysis is by cleaving vital proteins responsible for signal transduction like SNAP25 and SNAP-to-brevin. These complexes regulate acetylcholine release, which is responsible for muscle contraction. So just to break that down a little bit, here in figure A, you can see the normal condition. So what will happen is that the synaptic vesicle will bind to the presynaptic membrane, and then acetylcholine will be released and then bind to the receptors here, which will then cause your muscle to contract. In the case where BoNT is introduced, you see the cleavage of either SNAP-to-brevin or SNAP-25. So these are circled here in red. And then what you see is that then there's no binding to this presynaptic membrane. And then there's no release of acetylcholine. And when there's no acetylcholine release, it essentially means that there will be muscle paralysis. So the muscles will no longer, they will no longer contract. So as I just said, once SNAP25 or synaptobrevin is cleaved, the synaptic vesicle does not fuse to the presynaptic membrane, and acetylcholine is not released, and muscle contraction stops. Okay, so now we've established the background on BoNT's use, but what does any of this actually have to do with animal testing? So it's important to remember that BoNTs are biological proteins, making them distinct from chemically synthesized drugs, which are well characterized. Due to their natural origin, there's high level of variance. And due to this variability, they are required by law, as well as other biologicals like vaccines, to be tested on a batch-by-batch basis. In the European Union and the United Kingdom, among other countries in the Council of Europe, the European Pharmacopoeia sets out the standards for the quality of medicines in a series of monographs. BoNT A and B have their own monographs specifying product-specific quality control standards. In this monograph, which you can see here, which was last updated in 2022, the in vivo mouse test remains the standard method to test BoNT-A despite advancements in alternatives. So really briefly, the potency of BoNT-A and B are required by law to be tested by this in vivo, by an assay. And the most common assay, or the gold standard assay, which is referred to in the pharmacopoeia at length, is the MLB. And so a little bit on the MLB now. So the MLB, or Mouse Lethality Bioassay, measures the LD50, which is the dosage needed to cause mortality in 50% of the mice tested. For each test, around 100 mice are injected with serial dilutions of the test substance. After injection, mice are returned to their cages for observation for between 72 and 96 hours. During this period, mice are monitored for signs of botulism, which can include labored breathing, paralysis, and even mortality. A LD50 value is obtained by recording the lethality in each group and then developing a median LD50 using statistical methods. After the assay is concluded, all mice are euthanized. LD50 tests are scrutinized because they cause severe animal suffering as classified by the EU Directive 2010/63, which is on the use of animals in scientific research. Also, they show a lack of consistency between results. The MLB generally has several drawbacks. For one, they cause severe animal suffering, which is a very important concern to take into account. Also, generally, LD50 tests, as there's more than just the MLB, LD50 tests are a type of test. So there's more LD50 tests out there than just the MLB. But generally, these kinds of tests show a lack of consistency and are reported to, and I quote, extreme variability. Specifically, in the MLB, mortality is assumed to be directly related to asphyxiation from paralysis of respiratory muscles, but the toxin can generally impact muscular movement, preventing mice from accessing food and water, meaning they can sometimes perish from dehydration and or starvation rather than the toxin's effect alone. And death resulting from malnutrition is not accounted for in subsequent LD50 values, which could explain the inconsistencies with the assay. So really what this is saying is that they're not accounting for the fact that mice may die or they might perish from another reason than the toxicity of the substance causing death. Instead, it could be death coming from the fact that they can't access their food, they can't access their water. And so to put some numbers on this, the next bullet point here on the slide, you can see a 20% intra-laboratory, so intra is within the same lab, and a 50% inter-laboratory error rate when testing the same product batch. And so that, if that doesn't put off some alarm bells in your head, it should, because if you look at the number 50% inter-laboratory error rate, that's essentially a coin toss. So you're tossing a coin on whether or not, on how predictive this assay is, or rather how how consistent the assay can be. Also, species specificity is concerning regarding the translatability of the MLB. If you remember, I did state that there are different species-specific differences and how different species respond to the different serotypes of the toxin. Over the last 30 years, there are different new approach methods that have been considered for the potency testing of BoNT. Multiple review papers have been published with extensive overviews of both in vivo refinement and in vitro replacement methods. However, this lecture is mostly focused on non-animal methods, thus only in vitro assays are described. And in vitro assays are those that essentially you can think of in a petri dish. Furthermore, given our attention to the regulatory side of BoNT, I will focus on those in vitro methods that have been validated for regulatory application and or have been referenced by the European Pharmacopoeia. And two notable assays, which I'm going to discuss in further detail in the next slides, are the SNAP-25 assay and the cell-based assays. The SNAP-25 assay is an in-chemical assay measuring the cleavage of SNAP-25. So remember, SNAP-25 is that protein, and is detected using fluorescent labeling. Although this method sensitivity is similar to the MLB, it is not considered a complete replacement because it excludes multiple steps considered crucial for BoNT's mechanism of toxicity. And these steps are cellular binding, internalization, and intracellular activity of the BoNT protein complex. Various in vitro assays have been developed with the same underlying concept as the SNAP-25 assay, and these are all referred to as endopeptidase assays. Some of them even add microfluidics to increase assay sensitivity, but still some crucial mechanistic steps are missing. So enters the cell-based assays. So CBAs have become widely implemented and accepted as a full replacement for the MLB. The way the assay works is that they basically consist of neuronal cells, which are incubated with a sample of BoNT for 24 to 72 hours. Then the level of SNAP-25 protein cleavage products are measured. Various types of cell sources have been utilized, including primary neurons harvested from mouse embryos, continuous cell lines from humans or mouse neuroblastoma cancer cells, and stem cells derived neurons. Something I would like to note is that the neurons harvested from mouse embryos still require animal sacrifice. Also, neuroblastoma cancer cells are not typical cells, so they are not necessarily the best predictors either. And also, when you're using any kind of mouse cell, it's still not human relevant. So really what we'd hope to see is the use of the human material. To determine the potency of BoNT samples with a CBA, manufacturers use a reference standard, which is a sample of BoNT pre-calibrated in product-specific LD50 units using the MLB. So in short, it's a kind of control. Cell-based assays are considered a promising replacement because they can measure receptor binding, internalization, intracellular activity, and in some cases, the inhibition of neurotransmitter release. Also, various cell types and cell lines can be used, as I already said, meaning they can theoretically be performed using only human-derived materials. So now let's talk about when these assays are used. Production begins with bacterial and seedlot cultures, each checked for toxin activity using an acute toxicity assay. After purification, to remove harmful components, the bulk toxin undergoes a potency test. Next, the bulk toxin is diluted, mixed with excipients. So these are the things that make it a specific product. So they're going from the actual active ingredient to a product. So they mix different excipients in there, and then it's divided into multiple final product batches. And then each of these batches must then again be tested for its potency. Because every batch is tested and many come from the same bulk, batch testing accounts for the most animal use for MLB. So in summary, there are two stages, which are circled here. So this is during the bulk purified toxin here. And in the final product batches, when you see that the MLB or CBA will be used. And what I just said is that in this stage, during the final product batches, this is where you see the most animal testing because each one of these will have to be tested again. A sample of them will have to be tested again using the mouse or CBA test. So yeah, in summary, there are two stages when the potency testing must be performed. So now that you know a bit more about the technical details of cell-based assays, why is the MLB still used at all? Well, to understand that, I compiled a few benefits and drawbacks of the CBAs compared to MLB. So on one hand, CBAs have a lot of potential because they can use human material, for instance, stem cells. Products derived from human neurons can produce human toxicity values, which can aid physicians when converting from one product to another, because currently BoNT units are actually not transferable between products. Also, it can allow for species-specific potency evaluation. CBAs can also incorporate various important steps of BoNT toxicity and can be just as or more sensitive than the MLB because CBAs are designed on a product-specific level and have the potential to reduce intra-laboratory error. They also have a proven track record in regulatory safety testing and are even referenced in the pharmacopoeia. However, on the other hand, unlike the MLB, they are not transferable between products. So something to note here is that the MLB is actually transferable between products because the MLB can be used for all types of BoNT products. CBAs, as I already said, are going to be optimized per product. So they can't be used for different products, unfortunately. So, and the reason this is because each product contains its own excipients. So remember, if you remember back to the slide on the process, so the manufacturing process of BoNT, you saw the part where the excipients are added, and that's when the bulk toxins are no longer the same. Also, there's so much variability. So different manufacturers even use different kinds of BoNT in their products. So like I said, BoNT-B can also be used. And so therefore, the cell-based assays are not optimized for those specific products once the excipients are added. So each product has to be assayed in relation to its own reference standard. And that reference standard is calibrated using the Mouse Lethality Bioassay. And at present, the pharmacopoeia requires product level validation before market approval. Also, given that we're dealing with industry here, there are limitations in data sharing as the various CBAs are proprietary and may not be shared readily between companies. That's not to say companies don't work together, and in fact, they have worked together, but they don't necessarily work together all the time in a happy family, as we would maybe hope, because of course the assays themselves are going to be proprietary. So where do things stand today? Have manufacturers begun switching to the cell-based assay? And if so, which ones? And how far has this transition come? Because if you remember back to this slide, so here in 2023, this is 10 to 11 years after Allergan first received regulatory approval for their cell-based assay for Botox, the European Coalition Against Animal Experimentation was still working diligently to gain attention to animal testing for BoNTs, obviously showing that one company integrating cell-based assays is still not enough a decade later, and animals are still being used. In 2019, an opinion article was published on the use of mice for this purpose. And this article was published from the perspectives of animal protection organizations, so the European Coalition to End Animal Experiments, who I've been talking about already for some time. The article reported that Allergen, Merz, and Ipsen, which are all different companies, had validated cell-based assays for BoNT, yet the number of animals used for batch testing in Europe remained high. Although the authors expressed not all batch testing in Europe was attributed to BoNT, since vaccines and other biologicals are included, country data from Germany, Ireland, and the UK, which were identified through project summaries to still use the MLB for BoNT potency testing, showed no clear decline in mouse use. So if I could break that sentence down a little bit more, essentially what we're saying is that, or what they were saying in the article, is that Germany, Ireland, the UK were shown to be still using the mouse test when they were doing their research. And then when they looked at country-specific data, they did not see a decline in the number of mice used for batch potency testing, which could indicate, at least to them, that the MLB could still be used, as you would expect there to be a decline in mouse use if the MLB is being used in Germany, Ireland, and the UK. The authors also estimated that around 400,000 animals were used annually for BoNT-related batch testing across EU and the UK, and this is again from 2019. So this is data that's from quite a while ago, before Brexit, right? And though it's important to note that this figure is based on project summaries, which are so-called NTSs, or non-technical summaries, rather than final outcomes. So NTSs report data, basically what they intend to use, not what they did use. The paper also noted that BoNT's growing cosmetic use challenges the usual harm-benefit justification applied to medicines. And it called on regulators, so those who are involved with the European Pharmacopoeia, the European Medicines Agency, and the European Commission, to drive the transition by enforcing and rewarding non-animal methods. However, this study, which I'm just talking about, was published over 5 years ago, and no comprehensive update really exists on this issue from all these different perspectives, making this the first issue that I decided to analyze in my PhD. And so we set out to understand the current state of the art of the transition to in vitro replacements for MLB for BoNT batch potency testing in Europe. And we recently published our findings in the NAM journal. So if you're interested, I will also take like a 10-second little break here so you can screenshot or type furiously the name of the article. But also if you look at my name, I have not published many papers, so it's not too hard to find either. If you just look up Watkins and botulinum neurotoxin, should be one of the first things that pops up. But if you're interested, then definitely please check out this article. But in this paper, we aim to identify and describe the current use of in vitro alternatives for batch potency testing for medicinal use. Secondly, we identified market authorizations for BoNT and their relation to acceptance and implementation of in vitro alternatives. And last, we wanted to research based on the number of mice used for potency testing and the change in regulatory guidelines over time, if the transition to the CBA had changed much since 2019. In general, we aim to clarify current research gaps and highlight potential actions needed to phase out the MLB for BoNT potency testing from the European Pharmacopoeia using public data. So what did we find? Well, in short, we found that estimating the actual number of mice used remained really challenging and very uncertain. And even when we applied conservative assumptions and adjusted for project duration, the non-technical summaries, so remember I talked about them already, the summaries of the projects that are put before the project actually takes place, consistently overreported animal use compared to national statistics, which isn't surprising per se, because it is known that NTSs report a maximum number of animals, but still we did estimate what this would look like on an annual use by correcting for the project durations. So for instance, if we just turn our attention to one of these figures here, go to Ireland, year 2020, we estimated, based on the project summaries from Ireland, that about 117,000 mice were used for BoNT potency testing alone in 2020. And then when we compared that to the national uses of mice in 2020, we saw that it was 17% over-reporting. So what that means, or what that could mean, is that NTSs are either wildly over-predicting the number of animals they will use, or that in the UK and Ireland, the majority of regulatory potency testing using mice for batch safety testing, because this is a very specific number here, so I want to be as specific as possible because it can be a little bit difficult to explain without reading the paper, but this number here is the number of mice used for potency testing, batch potency testing, in the specific or specified country for that year. Okay, so again, batch potency testing can include other vaccines and biologicals, but still, if you're looking at the NTS numbers here, you'd still see an over-reporting by 17%. And so basically what you can conclude from that is, like I already said, NTSs are either over-predicting by a lot, or the majority of these mice attributed to potency testing is actually due to BoNT products for these two countries. And recently, I was told by a collaborator that UK authorities confirmed that it was fair to assume the number of mice used for potency testing here could be attributed to BoNT alone. Also, something that I think hopefully you noticed, I've mentioned Ireland, UK, and one other country, Germany, but Germany is not included on this table. And maybe you were wondering why, or maybe you weren't wondering at all, but you should have been, because actually what we found, to our surprise in a way, is that there were no NTSs from Germany which reported using the Mouse Lethality Bioassay for BoNT potency testing. And I inquired with someone from the statistical database, so the BFR database in Germany, what this could mean. And they suggested it's likely because BoNT is no longer tested using the Mouse Lethality Bioassay in Germany, which would be a great development. However, this is speculation. Though it's grounded, it should be taken with some caution. I am still a biologist after all, so there's everything has to come with an air of caution. So we also found that the European market now has at least 10 approved brands of BoNT represented by at least 5 market authorizers. So these are the 10 brands here up on the, I would say on the upper side of this timeline. And since 2019, Ledibo and Relfides, so these two here on the end, have been added to the mix. Even more interestingly though, as shown on the bottom side, so you're looking on this part of the timeline, 7 out of the 10 brands reported to actually have a cell-based assay developed and validated for regulatory use. Further, we have reason to believe an 8th product actually developed a CBA in early 2020, but it was not able to be confirmed with public data, so we didn't include it here on this chart. But so what does that mean for the transition? It seems companies are actually doing their due diligence, so why would animal testing for BoNT still persist? Of the 31 non-technical summaries we reviewed from the UK, Germany, and Ireland between 2014 to 2024, I'm going to use the laser pointer here again, you see that 19, so this group here, mention ongoing CBA development, and an additional 5 indicated that CBAs had been approved for potency testing by regulatory authorities. These 5 NTSs reported that they continued to use the MLB either as a preventative measure in case the cell-based assay breaks down, B, to adhere to international project licenses, or C, to ensure comparability between the CBA and MLB. So comparability assays essentially are a validation assay. And something I want to point out is this part right here, the international project licenses, because actually that was only stated by the UK NTSs, none of the EU ones. So it's also interesting to see if that is the case in the EU or not. NTSs that did not have an approved cell-based assay, at least yet, stated this was either because the cell-based assay was not yet approved for the qualification of reference standards, not yet approved for medicinal products, but may be approved for others, is undergoing development or validation, or is undergoing regulatory approval procedure. So this 5 here is the ones that have an approved assay already, and this 26 is the far majority which don't have an approved cell-based assay yet. Furthermore, we looked at the regulatory aspects of the transition. For one, it's important to highlight that Directive 2010/63 on the protection of animals states that in vivo tests are not permitted if an alternative is recognized in EU legislation. Compliance with pharmacopoeial monographs is legally binding, meaning alternative methods cited there should hold precedence over in vivo methods. With that in mind, we compared a past and current version of the pharmacopoeial monograph relating to BoNT and found the MLB is referred to as the standard method and outlined explicitly, while the alternative methods are only mentioned briefly. And this is in both versions. So in both the 2005 version and the 2022 version, they already mentioned these alternative assays. However, the newest version of the monograph does include a paragraph encouraging the use of alternatives. Still, and also I would also like to point out that the new monograph does talk about reducing harm, so they have some refinement options, but we also don't necessarily believe that refinement goes far enough. And still, even though the manufacturers are strongly encouraged to validate a replacement method, and it's stated as strongly encouraged in the text of the pharmacopoeia, it is not clear if there is any incentive or support on a European level for validation efforts. Additionally, during both versions of the monograph, they state that the MLB is used for new products and manufacturers can only retroactively validate an alternative assay. And this is highlighted in the following excerpt. After validation with respect to the LD50 assay, and in the interest of animal welfare, the product may be assayed by an alternative in vitro method that covers the mechanism of botulinum toxin activity. If I break this down, in short, that implies that to obtain an initial market authorization for BoNT-A products, the MLB must be performed before an alternative can even be considered. And this is further explicated by the following quote. No regulatory authority anywhere in the world will grant approval for a company to substitute the LD50 for an alternative assay, whether animal-based or not, unless they are entirely satisfied that the alternative is valid. But so what does this mean for the transition to CBAs, and is there a path forward? To summarize, we pointed out a number of insights, so things that we need to work on, but also some highlights, which are things that are going well. On one hand, poor transparency in public data made it very difficult to understand the number of mice used for BoNT testing and how this number has changed over time. And this number could have served as an indicator for how well the CBA is doing to replace the animal model, but the exact number remains elusive. Still, on the other hand, NTS data did show that Germany appeared to no longer use the MLB. Still, in the UK and Ireland, there is continued use of the MLB. But we have seen a positive shift towards CBAs by most companies. And though regulatory agencies require MLB for new BoNT products and for the qualification of reference standards, the EMA and other regulators have demonstrated an appetite for change, and public campaigning shows a portion of the population is not happy with the use of animals for this purpose. Still, cell-based assays remain product-specific, making it difficult for a global standard to be developed, but it's not out of reach. In our research, we found that in 2024, a project in Ireland is trying to close this gap. In short, this project aims to, and I'm quoting here, publish publicly available and free to use data proving that animals are no longer required for BoNT testing, end quote. They claim to essentially skip the use of the MLB already in the development of the product, which in theory would remove the need to prove potency in mice first and make the CBA the first step in potency testing rather than the last. It remains vague who is actually completing this project because NTSs are anonymized, but this sort of project paves the way for large regulatory reform should it be successful. So now I would like to conclude this lecture, or conclude my slides, with some takeaways and possible solutions. I think it's always great to end with a few solutions rather than just problems. So something that is important here is that the general public is likely not informed on the use of mice for BoNT potency testing, and it should be widespread information. Actually, this is something that someone actually mentioned in the short answer question. So that's great that you mentioned it because I'm about to mention it now too. So basically, in the European public, especially using this as an aesthetic treatment, may colloquially classify BoNT products as a cosmetic and not really think of them as being a pharmaceutical product. And in Europe, they actually have a ban on the use of animals for cosmetic products. So this is what the person said in the poll, but that people should be able to choose between products that use a cell-based assay versus a mouse lethality bioassay. Secondly, animal numbers for severe uses should not be so shrouded in doubt. There are avenues such as retrospective assessments where the actual animal use can be disseminated and shared just like NTS data. And another note here, retrospective assessments, which are done after the project, so they're the real numbers that were used, they're actually required for severe procedures, but not all member states publish their retrospective assessment analyses, which would be great to have, because then that would have been much easier than trying to make guesstimates on the number of mice. It would be much better to have the actual use. Also, companies switching to CBA should be well recognized, and efforts to switch the CBA or develop harmonized reference standards or assays should be funded and perhaps awarded. Lastly, the Commission has a duty to uphold the directive and could require timelines for phase-out and pressure member states that use the test to enforce these timelines and market authorizations. Further, the EDQM, who is in charge of the pharmacopoeia, should place BoNT as a priority for animal phase-out because this transition is so far along and has the potential to significantly reduce mouse use in Ireland and the UK. And these are just a few of the steps that could be taken. There's hope on the horizon for a future where mice use for BoNT is no longer required, at least in the EU. And this is an opportunity for Europe to serve as an example for the rest of the world that we no longer need to use severe animal testing for BoNT products, irrelevant of their use for aesthetic medicines or treatments for muscular disease.